All right, we're going to jump back in here and finish this up. So bioenergetics, how are our cells going to help make some energy for themselves, and what kind of laws apply to that? We will dive more into the actual minutia of the different processes, but this kind of gives you the background of kind of where these enzymes and cofactors kind of play a role in helping our energy producing processes. So bioenergetics refers to the flow of energy in living systems. Our first law of thermodynamics states that energy can be transformed, but not created or destroyed. So essentially, like if you think about like gas in your car, as you burn it, the energy turns your wheel, your axle, and makes your car go forward. So we're kind of transforming it from stored energy in that liquid form. We break down those molecules and generate combustion so that we can generate that energy. Now, most of our energy producing systems lose energy. So we have this degree of lack of efficiency. So most things aren't coupled 100% with that. So we do lose some energy along the way. Second law states that energy transformations increase entropy. Essentially, things get more disorganized. Every time we have a reaction to free up some energy, we're getting more and more pieces essentially, and harder to kind of recapture those molecules to make the energy storing molecule available again. And we have to have what's called free energy, energy that's in an organized state, primarily in the form of ATP, is what we're talking about there. If it's broken down, it's ADP and a phosphate, still got the stuff there, but it's no longer free. It's been broken down. So second law dictates all living organisms require continued input of energy, because again, we have this entropy factor where things get disorganized. We've got to keep adding it. So we need to feed ourselves every day. Plants need water and sunlight every day to some degree, depending on the plant. So that's kind of what that refers to. Plants can have the sun shine on it, obviously. They get photosynthesis, and that makes glucose for the plant in simple terms. If we're talking about like fruits or vegetables, things like that. And then we can eat that, and then we break it down to give us energy. So we're transferring it from the sun to the plant, and then from the plant to us or other animal species. All right, when we're breaking things down to free up some energy or trying to make new energy storing molecules, then we either have to have endergonic reactions take place or have exergonic reactions take place. And so when we are doing things like the Krebs cycle, and we're breaking down food, which is let's say glucose, which is the molecule that's shown up here at the top of this. If we have oxygen available and we have glucose, then we go through a series of steps where essentially we're freeing up and generating ATP all along the way as we slowly change that glucose molecule by having a bunch of enzymatic reactions. And then our final product is, well, we've created a bunch of ATPs, and then we've also made water and hydrogen and carbon dioxide, which are basically pretty safe waste products. We can blow off the CO2, hang on to the water, and we're good to go. So that's kind of the more typical, what we call exergonic reaction. We're releasing energy as we go, as we break things down. If we were doing an endergonic reaction, it means we have to level it up. We've got to put a little bit of power into it to store it, essentially. Many of those reactions involving ATP are going to be what's called coupled, so that we can try and improve the efficiency of going from the exergonic process back to the endergonic process, essentially. We want to try and keep things from just falling apart. We got to keep it fairly close to being able to regenerate the ATP, essentially. And so we have these they show the gears on this slide because it's meant to show kind of how the reactants drive the processes. So the big wheel, we've got the reactants, we're making the product, and that process, as we make that product, releases enough energy that drives the wheel in the opposite way so that we can take the reactants and then make more products that way. Again, we're making a different product in each of those situations, but we are going to recreate it as we get up there. So All right, there's ATP. Forming it is considered endergonic. We've got to take our ADP and we've got to put in some energy to add the phosphate back onto it so we can get ATP. And that's our final, like I say, that's the energy currency for the cell. And that's what our primary way is, that we're going to be able to split off that phosphate, release that energy, and then the cell can use that energy to drive whatever process it needs, any kind of active transport, anything that, any energy that's needed for making whatever product that cell makes, or for fueling muscle contraction, or sending a message in our brain, whatever the case might be. That's what we need that ATP for. So endergonic, because we've got to add some energy to basically recreate it, essentially. And so ATP is considered to be the universal energy carrier in all of our cells. We bring in food and we break it down. And as we break it down, we create water and carbon dioxide, but then we also create a lot of ATP. And it depends on which of the three processes we use to break that food down, how many ATP we're going to get out of the deal with that. All right, so a lot of what's happening when we're dealing with ATP and we're trying to use these enzymes and all these things, where we have a lot of what are called oxidation and reduction reactions that take place. And that's particularly true when we get to the what's called the electron transport chain, which is kind of like the final step in oxidative metabolism, which uses the Krebs cycle initially to generate a bunch of hydrogen ions, essentially, that our FAD and our NAD can carry, those enzymes that we talked about earlier. And just the exchange of ions is essentially the way that we kind of can change these molecules and release a little bit of energy along the way. So this is a little confusing, and there's a question on the test over these terms here that I always tell people to make sure you're clear on it, but a lot of times people still miss it. So electrons are negative. They're negatively charged. So if a molecule gains electrons, it is reduced. You've essentially like plus minus one. So if you have 2 plus minus 1, then you're down to 1, right? So it's reduced. If it loses electrons, it's oxidized. So if you lose a negative, then you become more positive. So if you're 0 and you lose a negative 1, well, now you're a positive 1. So it sounds weird. You're gaining electrons, but that means you're getting smaller from a charge standpoint. If you're losing electrons, then that means you're getting bigger from a charge standpoint, so you become oxidized. Now, where it gets confusing is these two terms here. A reducing agent is one that donates electrons. So it facilitates something else being reduced, which means it's the one giving up an electron, so it becomes oxidized. If we have an oxidizing agent, it's just the opposite. It's going to take on electrons, so it's going to get reduced, and it's going to make something else become oxidized. So an oxidizing agent helps a different molecule become oxidized. A reducing agent helps a different molecule become reduced with that. So those are all coupled reactions. If you're reducing one thing, you're oxidizing the other, and vice versa. So we transfer over those hydrogen ions typically, and our coenzymes that I mentioned earlier in the first set of presentation, the NAD and FAD are kind of our key ones that are involved as being able to bind with these extra hydrogen ions that we're producing, mostly as we break apart glucose to make it smaller and get down to the size that it needs for the Krebs cycle and the electron transport chain. So again, B vitamins are primarily where we get those NAD and FADs. So some people like this acronym, I guess, a mnemonic. Oil rig, oxidation is losing, reduction is gaining. So if that helps you keep it straight, just remember oil rig. And that doesn't apply to the agents, though. The reducing agents are going to be the opposite, because they're the things that are helping something else reduce or helping something else become oxidized. All right, so we do have certain molecules kind of like a reversible reaction that can go either way. It can give up some electrons or it can take on some electrons, so it kind of functions as a buffer a little bit. Oxygen is known as our key final electron acceptor. So when we look at oxidative metabolism, the way that we're going to try to maximize how many ATP we can get out of carbohydrates and fats primarily, if we have oxygen present, then it's going to help make sure that the process doesn't stop. If we don't have enough O2 present, then we can't, the NAD and the FAD get saturated with hydrogens, and we can't recycle those two things, the FAD and the NADH, and so then the earlier process kind of comes to a halt. So oxygen is critical for taking those hydrogens from the NADs and the FADs as they carry them over into the mitochondria. All right, so here's our two forms of NAD and FAD that are shown. And in the oxidized state, NAD is shown with a little plus, meaning it's already given up its electron, and it can take on one hydrogen. FAD is not shown that way. It's not shown as an ion. It's shown just as its normal self, and it's capable of taking on two hydrogens in that case. So it's a little bit different situation, a little bit more out of it. But essentially, as we're breaking down that sugar, they're going to grab onto some of those hydrogens that are being produced as we chop that glucose molecule up. So NAD gets 2 electrons and 1 proton. So again, it started out as NAD plus, meaning it was missing an electron. So it can take 1 electron, and then it takes on another proton and that electron too. So it ends up being shown as kind of this NADH typically, and then sometimes written with this extra H attached to it. FAD is a little bit better, and it stays more balanced as far as like not having spare charges left over. All right, the last video, I think. Let me stop that. And
cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
almost done. All right. So here's kind of our example of those processes that they were just shown in that video. So if we've got a given product that has this hydrogen attached to it, these 2 hydrogens, then it can transfer those over to the NAD, and it becomes reduced. So it's gaining these 2 hydrogens and the electrons associated with them. Over here, then it's going to dump it off, and it's going to become oxidized because it's going to donate those hydrogens over to this other compound, and then it can carry it on. And so we'll get into the electron transport chain, I think, the next lecture and talk about that. But there's just a whole series of reactions like that that take place to help us gradually pass those electrons along, and we tend to be able to generate some ATP along the way as we go. So I think that's it for today. I'm going to stop this recording and then try and get these posted so you guys can view them.